Hello students. So we are going to learn about gram staining technique today. So the aim of our experiment is to perform gram staining and identify gram nature of bacterial cells. The requirement for the experiment includes crystal violet stain, grams iodine, safranin, decolorizer, bacterial culture for performing the stain, and slides. Coming to the principle of gram staining. So briefly, before discussing about the gram staining technique, let us quickly revise about the structure of bacterial membranes in gram positive and gram negative bacteria. So remember, gram negative bacteria has an inner membrane followed by a very thin layer of peptidoglycan followed by the outer membrane. So overall, the lipid contents that are present in gram-negative bacteria is more. Gram-positive bacteria, on the other hand, they contain an inner membrane followed by the peptidoglycan layer or the cell wall layer, which is multi-layered. It doesn't show presence of an outer membrane. So overall, the lipid contents that are present in the gram-positive membrane is less. During gram staining, we make use of the four reagents. The first, we need to make the smear of the bacterial culture and add and heat fix it. Then we make use of the primary stain, that is crystal violet, which will enter inside the cell and will then we remove the excess dye and add the iodine that is acting as a mordant, which basically forms a crystal violet iodine complex. So here, both the gram positive and the gram negative will be stained blue at this stage. Now we do a decolorization treatment with either alcohol or acetone. Now on decolorization, the gram positive bacteria which have less lipids do not form pores in the membrane, and they have a very thick peptidoglycan layer as well that allows the gram positive cells to retain the complex of crystal violet and iodine. While the gram negative bacteria on decolorization, the lipids in the membrane they dissolve, reducing large pores in the membrane. As a result, the crystal violet iodine complex is lost. In the last step, counter staining is done using safranin dye, which is a pink color dye. So the gram-positive bacteria which have retained the primary stain, they will still appear purple in color, while the gram-negative bacteria which are decolorized after alcohol treatment, they will pick up the secondary stain, that is safranin. So this is the principle behind gram staining, and the same principle is written in detail in this slide. Coming to the protocol for this method. So firstly, we need a bacterial culture for which we want to perform gram staining. So we need to prepare a smear of the culture on the slide, followed by air drying and heat fixing of the slide. After that, we need to stain our slide with the primary dye, that is crystal violet, for at least 1 minute. After which we drain out the excess stain and flood the smear with iodine, which acts as a mordant. And here we continue staining for 1 minute. After which we'll wash the slide under a gentle stream of tap water. And then we'll perform decolorization using decolorizing agents such as alcohol for about 15 seconds. The slide is again washed under a stream of water and counter stained with safranin for 1 minute. The sides again washed under the gentle stream of water until no color appears into the appears out of the slide. Then we blot dry or air dry the slide, and using a stain of oil, using a drop of oil, we can observe the slide under oil immersion lens, that is under 100x magnification. Coming to the observation and results. So we can see that some cells will appear purple in color as purple or blue in color as seen in the image on the left. And these are basically gram-positive bacteria that have retained the primary stain, that is crystal violet, while the gram-negative bacteria, they lose the primary stain, crystal violet iodine complex, and hence they appear as pink in color. Also, this method is not only you will be able to identify the gram nature of the bacteria, but you can also identify different shapes of bacteria. Like you can observe bacilli in both of these images. Okay, bacilli are basically rod-shaped bacteria. This is the gram-negative bacteria. They are also bacilli, but they are basically short rods. So there are other shapes of bacteria as well, which can be identified by staining methods, including cocci, spirilla, and their different arrangements, such as cocci in pairs, chains, and so on. So thank you.